The Human In Vitro Drug Metabolism Dataset in the DIDB contains results from in vitro metabolism studies where a drug is tested as an inhibitor, activator, inducer, or substrate for a given human drug metabolizing enzyme and its variants. By typing a citation number in the search area, you will retrieve all collected data in DIDB documents containing this citation. Clicking on the citation will take you to the data tree view. Study results are organized according to the overall effect and mechanism of the interaction. A single click on a plus sign will expand the next layer of data, while double-clicking will display the full entry. For example, in this enzyme substrate entry, valsartan as an object is a substrate of CYP2C9, while in this enzyme inhibition entry, diacerin as a precipitant is an inhibitor of CYP2C9-mediated valsartan 4-hydroxylation. Metabolism parameters such as Km, Vmax, intrinsic clearance, Ki, and IC50, along with detailed experimental conditions, are curated. From this citation, you know that diacerin is an inhibitor of CYP2C9. If you are interested in all compounds that inhibit CYP2C9, you can do the following query. From this citation page, click Queries, then Enzyme. This will take you to the enzyme query page. Click on Precipitants Affecting Enzyme, find precipitants which inhibit CYP2C9, then click Submit. By conducting this query, you will obtain a complete list of in vitro inhibitors of CYP2C9, which includes 3,910 entries at this time. These results are presented in a table by default. You also have an option to view the results in a tree. In the table view results, you can customize the data for further meta-analysis. You can select or deselect columns. You can also filter the results to a specific substrate. For example, you can use the CYP2C9 index substrate, diclofenac, or select an object from the drop-down list. For inhibition parameters such as Ki, IC50, and K big I that have values presented in the unit of micromolar, you can use the numeric column to filter the results. For example, you can find all results with Ki values less than 10 micromolar. You can also perform an advanced table search where you can filter by one or several conditions. Additionally, you can rearrange columns and copy, download, or print the results. You can also learn how to best use the table features by watching the video here. Of note, by clicking on a citation number, you can go to the citation details of this entry together with all the other data entered under this citation, view the PK profile of the citation drugs, or go to the citation in PubMed or Semantic Scholar. In the DIDB, multiple queries allow you to retrieve an in vitro metabolism dataset by drug, enzyme, in vitro induction, in vitro parameters, or FDA markers. I invite you to check out our other video tutorials which provide in-depth looks into the contents and functionalities of the DIDB. And that concludes this video tutorial on the Human In Vitro Drug Metabolism Dataset. If you have any questions, just go to our website, druginteractionsolutions.org, and click on Contact Us.